Right. Our sixth talk is Shahin Nasr. It will be giving a talk entitled Differentiating the Impact of Amblyopia on Spatial Frequency Encoding within the Human V2, V3 Thin and Thick Stripes.
Thank you. I know everyone is hungry and me too. And let's just do it quickly. So thanks a lot for attending this talk. I'm going to talk about the impacts of amblyopia on activity within extrastriate cortex, including V2 and V3, and in mesoscale level. I'm going to talk about thin and thick stripes. So first, for those of you who are not familiar with amblyopia, amblyopia is a developmental disorder caused by unbalanced binocular visual input early in life. And there are two common causes for amblyopia. One is a strabismus, when one eye is misaligned relative to the other one, or anisometropia, when refractiveness in one eye is significantly reduced relative to the other eye. But don't let the term lazy eye mislead you. Technically, it's a brain problem. It's a developmental problem. And these eye conditions leads to, so they avoid visual cortex proper development. And amblyopia is technically associated with a very long list of visual impairments. And in this specific talk, I will talk about high spatial frequency encoding problems in these individuals. But for those of you who are interested in more high-level phenomena, please wait for the next talk. Dorsa will talk about that one. And our knowledge of neural disorders underlying this impairment, especially for spatial frequency encoding, let's be honest, it's relatively limited. And most of that knowledge actually comes from studies in non-human primates rather than studies directly in human. For example, here in this study that was done by Skersetat et al. In 1998, they showed that those neurons that respond to, in area V1, those neurons that respond to a stimulation of amblyopic eye, they showed lower optimal spatial frequency compared to those neurons that responded to a stimulation of non-amblyopic eye. But even when it comes to studies in non-human primates, most of those studies are focused on activity within first visual area V1, and our knowledge in subsequent visual areas, extrastriate visual cortex, like V2, V3, is even more limited. One of the reasons for that, other than technical difficulties, is that extrastriate visual cortex is an extremely heterogeneous area, and it consists of multiple neuronal clusters that they differ from each other in sense of their spatial frequency preference. For example, when you look at the area V2, you can easily see thin and thick stripe. This is still in monkey and a study that was done by Roger Tootell. And you can see that, thin and thick stripes, they show different selectivity for motion, color, and on top of that, they also show different selectivity for spatial frequencies. I will show data about that one. So this said, in this study, we jumped on studying these areas in mesoscale level. So we try to localize thin and thick stripe and then understand, impact of amblyopia on spatial frequency encoding in them. And more importantly, we are doing that directly in human in vivo. 11 participants, amblyopics, they helped us in this study. Among them, 5 of them was strabismic, 4 of them were anisometropic, and 2 showed mixed cases. Base corrected visual acuity varied between 2/40 to 20/300. 6 out of 11 of them, they were completely stereo blind, and 10 out of 11, they showed suppression of one eye, and the other person showed diplopia. And also 6 individuals with normal vision helped us in this study. We used high resolution fMRI, and we scanned our subjects in a 70 scanner. Resolution was 1 millimeter. And a stimuli consisted of luminance varying gratings, Spatial frequency varied between 0.1 to 5.79. Contrast level is either full contrast or relatively low contrast, 5%. An experiment was block design. Subjects did a very simple task of color change detection for the fixation spot, just to assure us that they are not asleep and they are attending to the screen. And each subject was scanned 2 times just for the main experiment. In each session, 1 eye was stimulated. And on top of that, to localize our regions of interest, we focus on, as I said, on V2 and V3. We localized thin stripe based on their color selectivity and thick stripe based on their motion selectivity. We also divided visual areas to central part and peripheral part based on subjects' own retinotopic mapping. Here you can see example from one stereo blind amblyop. You can see that, in area V2 and V3, motion and color selective stripes are completely interdigitated to generate a map like that. Subject was scanned 4 times. I'm not available. And then you can also see that if you don't have a spatial resolution, you're wasting your time. Because these stripes are completely interdigitated, and in absence of that resolution, you cannot differentiate them from each other. If you're interested in how we did that, what type of stimuli we used, what was the procedure, please watch the video. It's from VSS last year, and also preprint of our paper. But this one is just focused on controls and results. So I think, you can easily see here that, when we showed, low contrast stimuli in the color selective and motion selective, stripes, response to non-amblyopic eye relative to amblyopic eye showed a preference for higher spatial frequencies here relative to here, and also the same in motion selective areas. And as I said, this effect is more apparent when we show low contrast stimuli rather than high contrast stimuli. So here, when we show high contrast stimuli, effect is completely gone, and you see more or less the same, response to the stimulation of amblyopic eye rather than non-amblyopic eye. When we compare it relative to what we see in control, again, you can see no difference in controls. So it means that the difference that we see in amblyop is not the difference between dominant eye versus non-dominant eye. Is much larger than what we see there. Technically, we don't see that much difference between dominant and non-dominant eye in controls. But what about periphery? In periphery, you can easily see that a spatial frequency preference differs between color selective stripes compared to motion selective stripes. Color selective stripes, they prefer higher spatial frequency compared to motion selective stripes, especially when it comes to non-amblyopic eyes. When it comes to amblyopic eye, you see that shift in a spatial frequency preference just for within color selective ones. In thicker stripes, rather you see a general decrease in the level of activity, while the spatial frequency preference did not change between the two conditions. Again, when you make the stimuli high contrast, this difference is gone, and you don't see any difference between them. When it comes to low contrast, but in controls, again, you see no difference between the two eyes. However, there is an interesting phenomenon. Honestly, when we were writing the abstract, I had missed that. So I'm now, telling you this is beyond the abstract. We noticed that when it comes to the activity in dominant eye or either non-dominant eye, and we compare it relative to non-amblyopic eye, activity in a non-amblyopic eye is actually higher than what we see in controls. So technically, there is a sort of oversensitivity in a stimulation of non-amblyopic eye relative to what we see in controls. I really like to say that, oh, good, so it means that, their peripheral vision is better than our peripheral vision, a sort of, silver lining. But it's really hard to interpret this data. We need a lot of behavioral tests to show what is this phenomenon. Is it really a compensatory mechanism just because their central vision is not good, so they're shifting their resources to peripheral visions? I still don't know that, but this is the future steps for us. And to sort of, summarize the data and also give you a better chance to compare amblyopia versus normal vision, here you can see the response in amblyopic eye relative to non-dominant eye in controls. As you can see in the central part, non-dominant eye is much better or generates, a stronger response compared to what you see in amblyopes. And when it comes to periphery, this effect will be more limited to color selective stripes, and motion selective stripes, the difference is not significant. When it comes to non-amblyopic eye versus dominant eye, as you can see in the central part, there is no real difference. But when it comes to peripheral part, you can see a sort of a stronger evoked activity in response to stimulation of non-amblyopic eye relative to dominant eye in controls. And this difference is more apparent when you compare the response evoked in motion selective stripes. So in conclusion, we showed that there is a decrease in the level of response to a stimulation of amblyopic versus non-amblyopic eye when stimuli are low contrast, high spatial frequency. And especially in the peripheral visual regions, this effect is more selective in color selective stripes compared to motion selective stripes. And also in peripheral visual areas, when stimuli were presented to non-amblyopic eye, we detected an oversensitivity in motion selective neuronal clusters in response to low contrast stimuli. So at the end, I have to thank my team. Thank you, all of you guys, and especially Eric Geyer helped us a lot with recruitment and consultation. And as you can guess, these are very expensive experiments. Technically, it cost us something close to $15,000 per subject. And so thanks to our funding agency. And if you're in Boston area, if you can help us with recruitment, please contact us. If you're interested in collaboration, please contact us. And for younger audience, if you're looking for postdoc position, one of you sitting here in front, so please send me your CV. Let's have a chat. Thank you all.
Great. I think we have time for a couple questions.
Oh, yes.
Hi, that was very interesting. So the peripheral deficit you find, I suppose that you didn't find a difference between the strabismic and the anisometropic individuals.
I didn't have the population.
Okay.
So it's a very, right now we have only 11 subjects. It's really immature if I say anything. So I can't say.
From what we know from the psychophysics, we'd predict that it would be the anisomotropes that would show that.
Yeah.
In terms of your shifting of the peak spatial frequency, so why do you think that is?
I was hoping, still, again, because of the fact that I have just 11 subjects, I can't say that it's correlated with the depth of amblyopia. So it's directly related to that. I cannot say that. I see some, signs of that, but it still needs further.
But do you think it's simply relating it's reflecting the V1 deficit?
Oh, this effect is definitely larger than what we see in V1. So in that sense, you can say that, there are some, weaker forms of that in V1, but it's definitely larger and more exaggerated relative to what we see in the same individuals in V1. So effect appears to be getting stronger and stronger in the next areas.
So the question would be, and I'll leave this for a later talk, why that would be so.
Sure you don't have any more questions. I suspect I know what the answer is, but I have to press you anyway. You tested each of these subjects extensively, but you haven't told us anything about individual subject data. So surely you must have some idea about whether the tuning for each subject corresponds to their behavior. Whether you have subjects that range in acuity in the amblyopic eye from 2040 to 2300. Is there any association with those effects? Tell me what you can about them.
I wish I could say that. There are a couple of problems here. One problem is that, originally when we were doing these tests, we, in behavioral studies, we focus on central areas. And at this point, we still don't have the data from peripheral areas. So I really can't say anything about the correlation between behavior and fMRI activity. And on top of that, I said that, interpretation of data, or even I said that, my prediction, it's a very, very agnostic thing. I don't know, because sometimes activity goes up, and it simply means that these subjects are becoming more vulnerable to noise, low contrast noise. I don't know. This is ongoing. Thank you.
Thank you.